Hi everyone, I am Dr. C. S. Kulkarni, MD. I am going to teach medical biochemistry. Before starting the topic, please like and subscribe the channel. In the last session, we studied about nucleus, endoplasmic reticulum and Golgi apparatus. Now we will study further subcellular organelles. Lysosomes were discovered in 1950 by René Dewey, who was awarded with Nobel Prize in the year 1974. Lysosomes are tiny organelles. Solid waste of a township are usually decomposed in incinerators. Inside a cell, such a process is taking place within the lysosomes. In this diagram, we can see that there are tiny lysosomes. Endocytic vesicles and phagosomes are fused with primary lysosome to form the secondary lysosome or digestive vacuole. Foreign particles are progressively digested inside these vacuoles. Completely hydrolyzed products are utilized by the cell. In this diagram, we can see lysosome cell recycle center, which is a part of peroxidase recycle center. The lysosomes are a bag full of enzymes at an optimum pH of 5. As long as the lysosomal membrane is intact, the encapsulated enzymes can act locally, but when the membrane is disrupted, the released enzymes can hydrolyze external substrate, leading to tissue damage. Lysosomal enzymes have an optimum pH around 5. In this diagram, we can see that after the membrane ruptures, the cells undergo lysosomal dependent cell death, caspase dependent apoptosis, lipid peroxidation, reactive oxygen species dependent inflammasome formation and necrosis. The lysosome contain the following enzymes. These are polysaccharide hydrolyzing enzymes, then protein hydrolyzing enzymes, nucleic acid hydrolyzing enzymes, and lipid hydrolyzing enzymes. Thus, we can see that lysosomes are nothing but a bag of enzymes. Coming to lysosomal deficiency disorders, in gout, urate crystals are deposited around a knee joint. These crystals, when phagocytosed, cause physical damage to lysosomes and cause release of hydrolyzing enzymes. These lead to the inflammation and arthritis. Following cell death, the lysosomes rupture, releasing the hydrolyzing enzymes, which bring about post-mortem autolysis. Lysosomal proteases, that is catspin, are implicated in tumor metastasis. Catspins are usually restricted to the interior of lysosomes, but certain cancer cells liberate the catspin outside the cell. Then the catspin degrade the basal lamina by the hydrolyzing enzyme collagen and elastin, so that the other tumor cells can travel out to form distant metastasis. Silicosis results from inhalation of silica particles, which are taken up by phagocytes. Lysosomes membrane ruptures, releasing the enzymes. This stimulates fibroblasts to proliferate and deposit collagen fibers, resulting in fibrosis and decreased lung elasticity. Inclusion cell disease is a rare cell condition in which lysosomes lack the enzymes but they are seen in the blood. This means that the enzymes are synthesized but are not able to reach the correct site. It is shown that mannose 6 phosphate is a marker to target the nascent enzymes to lysosomes. In these persons, the carbohydrate units are not added to the enzymes. Mannose 6 phosphate deficient enzymes cannot reach the destination, which is a protein targeting defect. Coming to peroxisomes, in this diagram we can see tiny peroxisomes. The peroxisomes have a granular matrix. They are of 0.3 to 1.5 micrometer in diameter. They contain peroxidases and catalase. They are prominent in nucleosides and platelets. Peroxidation of polyunsaturated fatty acids in vivo may lead to hydroperoxide formation, that is, ROOH leads to ROO prime. The free radicals damage molecules, cell membrane, tissues, and genes. In this diagram, we can see halogenation cycle, which is a part of peroxidase cycle. In this diagram, we can see that ferric ions and glutamate undergo lipid peroxidation, which leads to ferroptosis. As we can see in the diagram, catalase and peroxidase are the enzymes present in peroxisomes, which will destroy the unwanted particles and other free radicals. Coming to peroxisomal deficiency diseases, deficiency of peroxisomal matrix proteins can lead to adrenoleukodystrophy or Brown-Schleider's disease, characterized by progressive degeneration of liver, kidney, and brain. It is a rare autosomal recessive disease. The defect is due to insufficient oxidation of very long chain fatty acids by peroxisomes. In this diagram, we can see that there is accumulation of very long chain fatty acids inside the cell, outside the mitochondria, because of their insufficient oxidation. In Zellweger syndrome, proteins are not transported to peroxisomes. This leads to formation of empty peroxisomes or peroxisomal ghosts inside the cell. Primary hyperoxaluria is due to defective peroxisomal metabolism of glycosylate derived from glycine. Here ends our this session. Kindly like, share and subscribe our channel. Thank you.